Cyanide poisoning inhibits the electron transport chain, blocking oxygen and stopping ATP production.
A miner was rescued after being trapped in a section of a gold mine where a fire had used up the oxygen. He was unconscious and had a faint smell of bitter almonds on his breath. At the hospital, his blood oxygen levels were normal after being given pure oxygen, but he remained in a coma. Doctors suspected cyanide poisoning from the smoke, which binds to cytochrome C oxidase in the electron transport chain. Tests were conducted on his muscle cells. Analyze the data table. Task, Part A. Analyze how cyanide poisoning disrupts the specific stages of cellular respiration in the mitochondria, leading to the observed cellular and systemic conditions. Part B. Propose a two-stage emergency treatment protocol for the patient, justifying each stage based on the biochemical disruption caused by cyanide. How cyanide poisoning disrupts cellular respiration. Cyanide blocks the last step of the electron transport chain. Cyanide binds to cytochrome C oxidase, which is the final enzyme in the ETC. This prevents electrons from passing to oxygen. No electron flow means no proton pumping. Without electron flow, the mitochondria cannot pump protons across the membrane. This causes the mitochondrial membrane potential to fall, as seen, low. NADH cannot release its electrons. Because the ETC is blocked, NADH cannot be converted back to NAD+. This makes the NADH and NAD+ ratio become very high, as seen in the patient. ATP production in mitochondria stops. The ETC normally drives ATP synthase to make ATP. This is why venous blood oxygen is high. The tissues did not extract it. When the ETC stops, ATP drops to very low levels. With no oxygen use in mitochondria, muscle cells depend on lactic acid fermentation for ATP. This produces very high lactate levels in the blood, as shown. Oxygen remains unused in blood. Because the ETC is blocked, cells cannot use oxygen even if it is present. This is why venous blood oxygen is high. The tissues did not extract it. Emergency treatment protocol. Stage 1. Give a cyanide antidote that removes cyanide from cytochrome oxidase. Antidotes such as hydroxocobalamin bind directly to cyanide and form a harmless compound. This frees cytochrome C oxidase, allowing the ETC to restart. Once the ETC is restored, NADH can be converted back to NAD plus an ATP production can begin again. Stage 2. Provide supportive treatment to restore oxygen use and correct acidosis, 100% oxygen, to maximize oxygen availability once the ETC is working again. Intravenous fluids to clear high lactate levels and correct the acidity caused by anaerobic respiration. As oxygen use returns, lactate levels fall, ATP increases, and normal cellular function returns. Thank you for watching. Check through the playlist for enjoy more videos.